In 2012, the United States saw the most cases of whooping cough in 5 decades. Last week, a study was done by the FDA, and it was published in the Proceedings of the National Academy of Sciences, where they looked at the effectiveness of pertussis vaccines at preventing whooping cough. Joining me on the phone is Tod Merkel, PhD. Dr. Merkel is a microbiologist and the principal researcher for this study in the FDA's Office of Vaccines Research and Review in the Center of Biologics Evaluation and Research. Hi, Dr. Merkel, and thanks for talking to me today.
Oh, hi. It's my pleasure.
Okay. Well, let's start off for my radio audience, with just some preliminaries, a little whooping cough primer. What is pertussis, and what are the symptoms that you typically see?
Okay, so pertussis or whooping cough is an infection of the respiratory tract of a bacterium called Bordetella pertussis. And so the bacteria infect most of the airway, but they don't get all the way deep down into the lungs. They infect the trachea and the bronchi and the nasopharynx. And the disease is characterized initially by what appears to be just a very simple cold, runny nose, watery eyes, and a mild cough. And over a period of a couple of weeks, that cough will become more and more severe until finally it enters what's called the proximal phase. And in the proximal phase, the coughing is very severe, very strong fits of coughing, and that last for 5, 6, 7, 10 coughs. And during that time, the individual who's coughing isn't able to take in a breath. So at the end of the coughing fit, they usually have a very strong inhalation, and that's where you hear this characteristic whooping sound, is when the people are trying to breathe in after the end of the coughing fit. So that's a pretty classic case of pertussis. And the coughing, that severe coughing, can go on for several weeks, and it's really debilitating for the individual who's going through that.
Sure. And certain populations are more vulnerable to this.
Absolutely. I mean, the most vulnerable individuals are children under 6 months of age, and the younger the child, the more vulnerable they are. With our current vaccine schedule, you don't even get your first vaccination until you're 2 months of age. So
right.
Absolutely most vulnerable kids are kids under 2 months of age. And there's two issues. One is, of course, they lack the immunity, but secondly, their airways are so small that they're much more prone to complications.
Right. And because this is caused by a bacterium, it can be treated with antibiotics.
It can be treated by antibiotics, but one of the problems with pertussis is, at least classical cases, they start out so much like a typical cold that parents don't worry about them at first. And one of the things that we've learned is that once the infection has established itself to a certain point, even if you eradicate the bacteria with antibiotics, the child will still go through a pretty severe case of pertussis. There's this point of no return. And often, because the disease starts out looking just like a mild cold, by the time the parent realizes this is something more than a cold and goes to the pediatrician, it's often beyond the point in which antibiotics are helpful.
Sure.
Antibiotics are still given at that point because if they're given soon enough, they will shorten the duration of the disease, and they will also prevent transmission to other people in the household. So it's still worthwhile, but this is one of the problems with the disease.
Right. Very good. And of course, vaccination is the method of prevention.
Right.
And I read your study, and the main point of your study, from what I could see, concerned vaccine effectiveness.
Right.
And you were looking at the difference of effectiveness between the whole cell and the acellular pertussis vaccine. First, I want to just, if you can describe the difference between the two. And you also mentioned there's concern about side effects with the whole cell vaccine, and if you could address that concern.
Right. So first, as you mentioned at the start of this, what we're trying to understand is why there is this increase in the number of pertussis cases. And this increase has been going on for quite a long time, and this is in the face of a very successful vaccination program. We vaccinate over 95% of kids by the time they enter school here in the United States. So the purpose of this study was to try and understand that. And one of our thoughts was that perhaps the acellular vaccine isn't protecting against infection as well as the whole cell vaccine did. And here we're distinguishing between infection, which is having the bacteria growing inside your airway, and disease, which is actually having the symptoms. And what we found in our study was that that in fact was true, that both vaccines, the acellular vaccine and the whole cell vaccine, protect against disease symptoms. None of the vaccinated animals got sick, but the acellular vaccine did not protect against infection. The vaccinated animals still had large numbers of bacteria in their airway for a long period of time.
Right.
And
go ahead.
The differences between the two vaccines.
Yeah.
So the whole cell vaccine was introduced back in the 1940s, and it is a killed whole cell, so that you take the whole bacterium, you treat it in a way to kill the bacterium, but the whole bacterium is there, so you're presenting all of these, all of the parts of the bacterium are present in that vaccine. And the acellular vaccine, what was done was 3 or 4 of what were believed to be the most important parts of the bacterium as far as initiating an immune response were purified. These proteins were purified, and a vaccine was made that consists only of these proteins and aluminum, which functions as an adjuvant.
Right. And like I said, you said there were side effect concerns with the whole cell, and that's one of the reasons the acellular was brought out. What are these side effect concerns?
Well, so the whole cell vaccine elicits a very strong response, and the children who are vaccinated with the whole cell vaccine have what we call site of injection reaction. So you have the arm will be sore. The kids generally run a mild fever. And those are the most common side effects. But of course, this also makes an infant very irritable, and anyone who has children knows that if your infant is unhappy, you're unhappy. Right,
but no severe side effects.
There are no life-threatening side effects.
Right.
But these were vaccines that were not tolerated well, and in the 70s and 80s, acceptance of this vaccine was going down because of these reasons. And I know when I talk to pediatricians who were practicing back in those days, all of them tell me that the whole cell vaccine is really not an acceptable vaccine for us to go back to because of these side effects. So the purpose of this study was not to suggest that we should go back to the whole cell. The purpose of the study was to understand the differences between the two vaccines so we can design, hopefully, a better acellular.
Okay, and let's get to the study itself. You've already addressed some of it, and you conducted it. You tested these vaccines on non-human primates, baboons to be specific.
Right.
And you set up 2 groups of baboons and tried the different vaccine on each group. And specifically, what did you find?
Well, so specifically what we found is when we vaccinate, well, first of all, just to set up, one of the earlier experiments we did was to take animals and give them pertussis, and then after they recovered, wait 6 months, and then try to give it to them again. And we were unable to do that. In that situation, we never recovered any bacteria from those animals the second time they were infected. So the initial infection gave them a type of immunity or an immune response that completely prevented them from reinfection. And what we did in this experiment was we took animals and vaccinated them with the whole cell vaccine and animals and vaccinated them with the acellular vaccines, and then we challenged them, tried to give them pertussis. And what we found was with the acellular animals, the acellular vaccine did an excellent job of protecting against disease. We saw absolutely no sign of disease in those animals. But when we went into their airway and tried and looked for how many bacteria were there, we saw that the vaccine had little or no impact on the number of bacteria in the airway or how long the bacteria stayed in the airway. And in contrast, the whole cell vaccine, there was an initial infection, but those bacteria were cleared in 2 to 3 weeks. The acellular vaccinated animals didn't clear the infection until 5 or 6 weeks. So the next thing that we did was, since we had seen that acellular vaccinated animals were infected and carrying the bacteria around, we wanted to know whether or not they could transmit. And so we took acellular vaccinated animals after they were infected, and we co-housed them with animals that weren't infected. And in fact, they did transmit the infection. So we showed that not only are acellular vaccinated animals infected, but they're capable of transmitting to individual animals that they come into contact with.
And the use of baboons in this study is because they exhibit very similar symptoms as humans do.
That's right. I mean, one of the problems with working with pertussis is it is very highly evolved and adapted to infect humans, and it doesn't infect other animals the way that it does humans. And so the baboon model was the first model where we were able to show really the classic symptoms of pertussis, including transmission to other animals.
Right. And so what do you conclude?
Well, so our conclusion is that the acellular pertussis vaccines are very effective against disease. I mean, we know that from clinical efficacy studies. We know that from use in the field, actual real use in the field. And our study just confirms that. But what our study does show is that the acellular vaccine does not protect. Again, this is assuming that what we've seen in baboons can be translated to humans. But right. If that's the case, then what this is telling us is that the acellular vaccine is not doing as good a job as the whole cell did in controlling the spread of the bacterium through the population. So although people aren't getting sick, they can still carry the bacterium and they can still transmit it. And we think that this can explain the increase we're seeing in the number of cases, because if the bacterium is moving more freely through the population, then the probability or the likelihood that a susceptible person is going to encounter someone who has the bacterium is higher.
Well, I have to say this is a very interesting study and a very useful study too. Let me close, Dr. Merkel, with one last thing. Is there going to be any additional research with you and your group on this topic?
Well, right. So yeah, absolutely. So this is really just the beginning for us. I mean, we think we have an idea now of what's going on, and the goal is to try to identify. We know that in a natural infection, you get an immune response that prevents you from being reinfected. And the whole cell doesn't do that as well as natural infection. And the acellular vaccination doesn't do it as well as the whole cell. So the goal here is to identify what exactly is that immune response that's giving that very good protection. And if we can add that to the acellular vaccine, we would then have an acellular vaccine that not only protects against disease symptoms, but also prevents infection.
Great. Well, I've been talking to Dr. Tod Merkel, a microbiologist with the Food and Drug Administration, concerning the pertussis vaccine study. Thanks, Dr. Merkel, for your time and expertise. I really appreciate it.
Oh, my pleasure. Thanks for calling.
Okay. Take care. Bye.
Bye.